Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Joint class. Dear student, we are in a continuation of the chapter that is plant growth in development, and today we are going to discuss one of the important topic of this chapter, that is seed dormancy. What we are going to understand seed dormancy. So let's understand what it is first. So we know that seed dormancy is a suspended growth of the seed. Seed is not able to grow, germinate now due to many factors. Okay, and we have discussed about the hormones that germination of a seed is carried out by the gibberellic acid, whereas the inhibition is done by the we can say the ethylene and the abscisic acid. So seed dormancy, specifically abscisic acid, seed dormancy, it is a state in which seeds are prevented from germinating even under a suitable condition. Means environmental condition are safe, they are good, then also seed is not able to germinate. Still seed is not able to germinate, it is getting prevented and not in germinatory in a suitable condition, then it is known as seed dormancy. Why it is there? Because in suitable condition also it is not growing, because there should be some defect or some problem in the internal factors. So if a suspended growth is there or the seed is not germination due to internal factor, then we call it as a dormancy. So here we have to distinguish two different situations, and seed physiologists have used two terms. One is quiescence and one is dormancy. Actually, quiescence is the condition of a seed when it is unable to germinate only because of the favorable external condition. Okay, so seed is unable to germinate only because of an unfavorable external condition. Normally required for the growth are not present. Means a favorable condition, because the favorable external condition which is required for generally for the normal growth that are not present, means the surrounding condition are unfavorable. And due to the unfavorable condition or environmental defect, the seed is not able to grow, then it is known as quiescence. But the dormancy is the condition of a seed when it fails to germinate because of internal conditions. Even though external conditions are favorable, means favorable conditions are there, external factor are good, but still there is some defect in the internal factor, and due to which it is unable to germinate, or due to which it is unable to grow, then this is known as dormancy. So dormancy is due to the internal factor and quiescence is due to the external factor, in short. So external factor can be temperature, moisture, atmosphere. They cannot be proper. In improper condition, that can happen. Seed may remain viable but unable to germinate to grow for several reasons. There may be different reasons for the seed not able to grow, and this can be on external or internal conditions. So it may be the, if we talk about the causes of seed dormancy, then hard seed coat is responsible. Okay, so if we talk about regions of dormancy, then the hard seed coat it is there. So after this, impermeable seed coat is there. Then immature embryo can be there. Germination inhibitors can be there. These all are the possible regions for the causes of a dormancy or regions of dormancy. So an internal situation that is easy to understand is an embryo that has not reached the morphological maturity capable of germination. So first region, if we discuss, that is the immature embryo. So embryo is immature at the time of seed shedding, and the seed will remain dormant till the embryo become mature. And it is seen in case of the ranunculus, that is due to the immaturity of the seed. Okay, so germination now does not occur due to the immaturity of seed. Then it is one example is the ranunculus. Now the dormancy of seed may be caused by a hard seed coat that prevent uptake of oxygen or water. If there is a hard seed coat, so due to which the seed, we can say due to is a hard seed coat can be said as a mechanically resistant, and it does not allow an embryo to grow because of the deficiency of the water and the oxygen. Then, so this is the oxygen and that cannot enter. So that can be the region. Okay, apart from this, if we talk, there may be the impermeable seed coat. there may be a impermeable seed coat. So impermeable seed coat for water and gases. So water and gases cannot reach the particular embryo, and due to which also the germination cannot occur and seed dormancy continues. So if we talk about some of the example of the impermeable seed coat, then it can be seen in case of the apple and chenopodium. Okay. Apart from this, the another is the after ripening treatment. This may be needed for the germination to continue. So sometimes seeds require a period of storage, a period of dry storage for developing the ability to germinate. So this the requirement of a period of a dry storage for the development of ability to germinate, then it is also one of the phenomena or after treatment that can be needed for a germination to continue. So such things are observed in case of wheat, oat, barley. Okay. Apart from this, the another region which is the important one, that is the germination inhibitors. If you talk about the germination inhibitors, then they occur in the seed coat and cotyledon of the embryo. This germination inhibitor occur in seed coat and cotyledon of embryo. So this germination inhibitor, as we have studied now, just they are found in the particularly cotyledon of the embryo, or we can say the seed coat. Okay. Now the important germination inhibitor causing the seed dormant. So examples of germination inhibitors are: number one, abscisic acid; number two, phenolic acid; number three, ferulic acid; number four, coumarin; number five, short fatty acid; sixth, cyanogenic chemicals. These all are the particularly we can say one type of, we can say, the germination inhibitors. So these all germination inhibitors are specifically found in this seed and responsible for the inhibition of seed germination or the dormancy. Now, now breaking of seed dormancy. How the seed dormancy can be broken. So if we talk about the breaking of seed dormancy, first we talk about the natural phenomena. Then in nature, seed dormancy is broken automatically. So first, natural way of breaking seed dormancy. If we talk about a natural way of breaking a seed dormancy, then naturally it is broken automatically naturally it is broken automatically due to number one, that is the development of growth hormones, development of growth hormone to counter growth inhibitors, to counter growth inhibitors. So naturally, obviously, it is going to break by the development of growth hormones if take place, which can counter the effects of the growth inhibitors. Second way is maturation and after ripening of embryo. Maturation and after ripening of embryo. Third, weakening of impermeable and tough seed coat. Weakening of impermeable and tough seed coat by microbial action, aberration, and passage through digestive tract, etc. Okay, so this many thing can be there through which the naturally the seed dormancy can be broken up. Now, next we are going to talk about the artificial way of artificial way of breaking seed dormancy. When we talk about artificial way of breaking the seed dormancy, the first thing that done in an artificial way, that is a scarification. And if we talk about a scarification, scarification means the hard and impermeable seed coat, hard and impermeable seed coat is weakened or ruptured by filling, chipping, hot water, and chemical treatment through acid, and by using knife also a scarification can be done or scar can be created on this particular seed coat. The second way is stratification. In stratification, seed are moistened, and exposed to to oxygen and exposed to oxygen for a for variable period at low and high temperature. So by moistening and then exposing to oxygen for a variable period in a hot and low temperature, then by such treatment, the seed can germinate. Then third way is vigorous shaking and vigorous shaking and hydraulic pressure are used to vigorous shaking and hydraulic pressure are used to weaken seed coats. Then D is counteracting inhibitors. Now in counteracting inhibitors, it is known that inhibitors are destroyed. Inhibitors are destroyed inhibitors are destroyed by dipping seeds in KNO3, thiourea, KNO3 thiourea, ethylene chlorohydrin, ethylene chlorohydrin, then gibberellin. So this some particularly important component which can counteract the inhibitors. Now, so this is the we have discussed the regions of dormancy and how the dormancy can be broken up. Now, next we are going to discuss the importance of seed dormancy. When you talk about importance of seed dormancy, the first we talk that is perennation. Means seed dormancy allows seed to pass through allows seed to pass through unfavorable conditions like drought, cold, etc. Then second is the importance of seed dormancy is dispersal. In dispersal, obviously, it is essential for the dispersal of seed. essential for dispersal of seed. Third importance is seed germinate only when sufficient water is available. When sufficient water is available to leach out inhibitors and soften the seed coat. Okay. So for this different regions, we can say the particularly the seed germination can be important. And the fourth importance is it helps human being to store, it helps human being to store grains, pulses, grains, pulses, and other edibles and other edibles for making them for making them available for making them available not for throughout the year and transport to the areas of deficiency and transport to the areas of deficiency. Okay, so such thing can happen through this particularly what a seed dormancy. So such a way seed dormancy is important. Now the question is, if seed dormancy is occurring, then how the germination will occur? So next we talk about seed germination. So it is a removal of seed dormancy or breaking of seed dormancy, that is nothing but simply a seed germination. So now the question is, how this seed germination take place. So it takes place in three different ways. We are going to discuss here three type of germination. First, epigeal germination. Then hypogeal germination. Then viviparous germination. And we call this viviparous germination as vivipary also. Okay. So we are going to discuss the three, how they take place. Okay. So first, we are going to discuss the epigeal germination. In this germination, hypocotyle grows first. Okay. So hypocotyle grows first means what? As you know, in embryo, like this, the cotyledon, this is embryo. This is the plumule, this is epicotyle, this is cotyledon. Actually, this is a whole embryonal axis. So the region of embryonal axis between the plumule and the cotyledon attachment site, that is known as epicotyle. Same way, this is the radicle, and the regions of embryonal axis between the cotyledon attachment site and the radicle, it is known as hypocotyle. So as you know, hypocotyle is the basal portion. So if hypocotyle grows first, it will push this cotyledon to come out of the soil like this. If this is the soil surface, and this is the seed, and this seed have this particular cotyledon and this thing. If this grows first, then what will happen? The cotyledon will initially remain this side, and then cotyledon will come out of this side. Okay. So if this way the germination will take place, cotyledon comes out of the side. Okay, because hypocotyle is going to push the cotyledon to go out of the side. Okay, then next is the viviparous germination. Hypogeal germination. In hypogeal germination, epicotyle grows first. Epicotyle grows first. And as epicotyle grows first, the cotyledon remain within soil. As you can see, this is the surface and this is a seed, suppose. If this is going to grow, the cotyledon are going to remain within the soil. Okay. So this way the cotyledon remain within the soil during the hypogeal germination. Now, the third type which we are going to discuss here, that is the viviparous germination. Third type is viviparous germination. So viviparous germination is a germination of seed within a fruit over a plant. Now the question is, why this germination is taking place over the plant within the fruit itself without falling on the seeds? That is the important question. So because suppose this is the particular plant, and here there is a salty area. So if generally fruit will fall over this salty area, the exoosmosis occur and the seed is not going to germinate. So to avoid this condition, so if this continuously fruit is going to fall or seed is going to fall on this salty area, it is not going to germinate, or it will not germinate. Why it will not germinate? Because the salty area will not allow this to germinate. So the evolution occurred and this seed started germinating over the plant within the fruit. So this germination is known as what we can say the viviparous germination. And it is seen in case of mangrove plants like Rhizophora, Avicennia, Soneraceae, and Heritier. Okay, so in this particular, we can say plants, this the germination occur of a seed over occur or take place over the plant, okay, within a fruit itself. So that is we can say the one important thing with respect to the germination. So this is all particular types of germination, and that is all about what a seed dormancy and germination. So the process in which seed coat is removed or injured mechanically, that is obviously a scarification, because scar we are going to produce over that. Then which of the following correct about seed dormancy? Hard seed coat results in seed dormancy. Yes, it is impossible to remove seed dormancy. It is incorrect. Seed dormancy helps the seed to survive in unfavorable condition. Yes, both 1 and 3 are the correct about the seed dormancy. Okay. Next, hard seed coat that prevents uptake of O2 and H2 can be seen in. So that is obviously in case of Fabaceae. Presence of chemical instead promotes dormancy in tomato. Ferulic acid. In tomato, ferulic acid is present that inhibits the germination. In which of the following plants the seed will not germinate until they are exposed to low temperature? Obviously, it is a polygonal. You have to remember this example. Okay, so that is all about what seed dormancy and seed germination. I hope you understood that. Thank you. Thank you, everyone.